Hi students, we have to proceed the lesson that is chromosomal basis of inheritance further. Now, today we have to talk about the comparison between the gene and chromosomal behavior. I already mentioned there is a parallelism there is a similarity between the behavior of chromosomes and Mendelian factors. Now here, we are taking the chromosome, how far the chromosomes are arranged. You know that one, all around the world, the scientists have established the total number of chromosomes is generally constant in all the cells of a species. If you take one particular species, the number of chromosomes is always constant for a particular species. The highest number you can see in the case of Ophioglossum, about 1262. And the lowest number of chromosomes in plants we have Haplopappus, gracilis, the golden what we have the slender golden weed. So the chromosome number is constant for all the cells of a species. Now if you are taking the diploid karyotic cells or diploid karyotic organisms, they have two haploid set of chromosomes, n plus n. One from each parent, one set from male, one set from female. Now the behavior of chromosomes during meiosis not only explains the Mendel's principles, but also leads actually new and different approaches about the heredity. So we are studying the behavior of chromosomes. It provides more and more information to study about the heredity. Now here representing what we have three pairs of chromosomes. Chromosome 1, chromosome 2, chromosome 3. So in each one a pair of chromosomes are present. And each pair is called a homologous chromosome or a homologous pair. Out of the two chromosomes in a pair, one chromosome is coming from a female parent called maternal chromosome, for example the black. And another one is coming from what we have, that is the male parent, for example the red one. That is called the paternal chromosome. So a homologous pair is normally formed of one that is what is called maternal chromosome, another one a paternal chromosome. The arrangement of genes, the sequence of genes, and also size, shape, everything is normally similar in both the chromosomes. So, if you are analyzing the nature of arrangement of genes, the genes are arranged in a linear order. That means we have the same genes in same order. I represented in a chromosome. We have the same genes which are arranged in the same order. Suppose chromosome, point number 1, point number 2, point number 3, like that. Here represented a pair of genes. So this one actually homologous chromosomes. But the genotype is heterozygous. One dominant allele, one recessive allele. Let's assume this is point number 6 or point number 10, any point. In that point, we have the same gene, A gene. It may be a dominant gene or recessive gene. Generally, we can say same genes in both the chromosomes. That is A gene in point number 10. Maybe you have B genes in point number 1, capital B or capital B small. So in a homozygous condition, we have two dominant genes or two recessive genes. In a heterozygous condition, we have A gene in different form of dominant and recessive. So you see that one take a pair of chromosomes, homologous chromosome, the chromosome number 1, where we have the gene A and A representing the heterozygous condition. These two are homologous chromosomes. I am representing two more chromosomes, chromosome 2, chromosome 3. So these two are homologous chromosomes, and these two are homologous chromosomes of what is called the third pair. If you are comparing one member, what is called homologue of one pair and the homologue of another pair, these two are called non-homologous chromosomes. So the member of one pair and the member of another pair, they are considered as non-homologous chromosomes because they are different from each other, either in size or in number of genes and the arrangement of genes. So if you are taking a pair of homologous chromosomes, they have same genes. arranged in the same order. This is number 1. Here I am taking for example a dominant gene, recessive gene. That is allele A, that is what is called dominant allele A and recessive allele A. If you are analyzing the nature of the genes, I mentioned already the genes are arranged in the same order. So same gene, same order in both the chromosomes. But here it is a dominant gene and a recessive gene. But they are differing from each other, namely allele A, that is in DNA sequences. Though the two chromosomes are similar in having same genes arranged in same order, if you are analyzing the nature of the DNA, they are different in the DNA sequences. Suppose you are taking allele A. I represented the base sequences. You see GCA like that. There are 23. Base pairs are represented. For convenience, you can write any number of bases. In the case of allele A, suppose you are taking for example position number 12. This is what we have. We have the base that is what is called C, the nucleotide C. If we are taking the recessive allele, so this is because of mutation, we have a gene that is, the base T, thymine. So there you have what is called cytosine nucleotide. Here we have thymine nucleotide. Because these two are actually alleles of what is called a factor A. So they are differing from each other. If both are dominant, we have here also C, here also C. But this is a dominant allele and this is a recessive allele. So the base sequences, I mentioned here the different DNA sequences. Actually, the base sequences is different between dominant allele and a recessive allele. So that's about what we have the diagram showing the behavior of chromosomes. Though the genes are A, they are different from each other in heterozygous condition. In all cases, if you are comparing the DNA sequence, the DNA sequence is different from, that is dominant allele A from recessive allele, at least in one base. There I represented C and here I represented T. What we have in the 12th position. What we have in the 12th position or the 13th position. Anyway, we can represent any sequence, but you have to represent one base at least different from each other. So, if you are checking this recessive and the dominant alleles, they have same gene, same order in the homologous chromosomes. If you are considering different genes, they have same, that is genes, they are arranged in the same order. For example, the first gene is, for example, capital B and the C, like that. This is all called the order. Same order. Same genes also, A gene, B gene, C gene, like that. If you have A gene here, you also have A gene. If you have B genes here, you also have B gene. Either in the form of dominant allele or in the form of recessive allele. So this is one comparison between gene and chromosome behavior. So the genes are different in their behavior. That is, in that case, what we have dominant and recessive alleles. But the chromosomes behave same, have the same genes, arranged in the same order. Now let's see the comparison between what is called the chromosome behavior and Mendel's factor in the form of statement. Now let's see the parallelism between Mendel's factors and the chromosomal behavior. There is nothing with the similarities between the Mendel's factors, nothing with the genes or the factors we can see, and the chromosomal behavior during what is called the cell division. Now if you are observing the alleles of what is called a factor, for example, A gene, so the alleles occur in pair. For example, if you are taking this chromosome, a pair of homologous chromosomes, now we have the genes in example of B. So the alleles of a factor, so factor B, the alleles of a factor, suppose I am taking capital B and small b instead of homozygous condition, they occur in pair in a homologous chromosome. Same is the case in the case of, for example, chromosome behavior. The chromosomes, for example, homologous chromosomes occur in pairs. There is one similarity. So, during gamete formation, either the similar or dissimilar allele. Now, similar allele, for example, capital A, dissimilar allele, capital A, small a. So, the similar or dissimilar allele of a factor, for example, A factor, separate during gamete formation. so that each gamete receives only one of the two factors or one of the two alleles, we can see. So the similar or dissimilar factors separate during gamete formation, so that each gamete receives only one of the two what is called the alleles of a factor. That is number 2. And also now the genes are located on the chromosome. The meaning for what is called the segregation of genes is nothing but the segregation of chromosomes. Only when the chromosomes segregated, the genes also segregated, entering into different gametes. Because the chromosomes are the carriers of the genes. So the second point, the homologous chromosomes normally separate during gamete formation. Then only we can have each gamete receives only one of the two alleles. This is because the chromosomes are the carriers. That's why the behavior of the chromosome is similar to the behavior of what we call the Mendelian factors or the genes. Now the Mendelian factors are sorted out independently. The meaning for that one, there is no connection between the separation of first pair of chromosomes to the second pair of chromosome. Each and every pair of chromosomes actually are sorted out, segregate independently without affecting the segregation of other factors or other what we have the chromosomes. So the Mendelian factors are sorted out independently. That's why we have received 4 different types of gametes during diabetic class. This is because of independent assortment as per the Mendelian principle. But mainly it is because of crossing over, we'll see later. So according to the Mendelian principles, what we have, the factors are sorted out independently. There is no connection between 2 different factors located on different chromosomes, or we have 2 different what is called homologous chromosomes of the same pair. So they are sorted out independently without attacking, without following others, what is called segregation. The same manner the paired chromosomes also segregate independently during meiosis, just like what we have the genes. Then only we have the different what we have the gametes. But the genes present, what we have the linked genes present in the chromosome, in the same chromosome, they do not assort independently. When the genes are present on different chromosomes, then only independent assortment takes place. But when the genes are present in the same chromosome, the linked genes do not assort independently. Only the chromosomes assort independently, but the linked genes do not assort independently. We'll see later about what we have the linkage phenomenon. So this is the parallelism or the similarities between the Mendel's factors and chromosome behavior. Occur in pairs, they also occur in pairs. Now they separate during gamete formation, they are also the chromosomes separate during gamete formation. They are sorted out independently without affecting other, without knowledge of other. There also the chromosome, the homologous chromosomes are sorted. The meaning for the one segregate, separate and enter into different gametes. Okay, they are following their own way, not following others' way of chromosomes. So that's about what we have the parallelism between Mendel's and chromosomal, Mendel's factor and chromosomal behavior. I will list out the copy of that one in the what is called the video. Okay. The important aspects to be remembered about the behavior of chromosomes during cell division, that is particularly meiosis. We have already studied meiosis. So what are the events normally took place normally during meiosis? We have meiosis 1, meiosis 2. So in meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Likewise in the case of meiotic division 2, prophase 2, metaphase 2, anaphase 2, and also we have the telophase 2. Now you have to recall those events. So what is the first aspect? The alleles of a genotype. The alleles of a genotype occur in the same locus of homologous chromosome. This is point number 1. I am taking the alleles capital A, small a. They are located at the same locus. Say an example I mentioned already. Suppose this is the 10th locus. So the alleles of a genotype located at the same locus in a homologous chromosomes. That is number 1 point. Now the second point, during S phase or what is called interphase, the synthetic phase. So during which what is happening, the replication process takes place. So the replication process occurs only during the S phase of interphase, the synthetic phase. So during the phase of what is called interphase of meiosis, what will happen? Each chromosome duplicates or replicates. So that forming actually each gene into two copies, forming two alleles or two copies for each allele. You see, because of the separation or the splitting duplication, we have two alleles for a fact. So in each chromosome there are two alleles because of the duplication or the replication of the chromosome, and so also the genes. So we have, you see that one, two capital A genes and two small a genes in a pair of homologous chromosomes. Now during anaphase 1, what is happening? Just in the first cell division, meiotic division. Now the two homologous chromosomes separate. The two homologous chromosomes separate. So that what is happening? We have two different alleles. The separation of two different alleles also having two different alleles. So this is one chromosome. Now we have haploid set in anaphase 1. Just normally see that one. We have what is happening in anaphase 1. Now the chromosomes are moving away from each other. As a result, we have the separation of the two chromosomes resulting in two different alleles. Okay, this is one pair of alleles, another pair of alleles. Here I am using the word allele referring to the genes. So each one is having the sister chromatids. So now we have the cells in anaphase 1 have half the number of chromosomes. One with, for example, if you have 26 chromosomes to start with, one end we have actually in anaphase 1 just 13 chromosomes like that. So what happens during anaphase 1? The homologous chromosomes separate so that each cell has only, that is what we have, or each, just normally we can say the cell, I'm using the word cell, so that what is happening, we have two different what is called the alleles, the separation of the two different alleles, so that we have two different what is called the alleles of a factor. Now during anaphase 2, the sister chromatids undergo separation, the separation of sister chromatids of the homologous chromosome. Because of the division of the centromere, now the sister chromatids get separate. These two are called sister chromatids, these two are called sister chromatids belonging to one homologous chromosome. So during anaphase 2, the sister chromatids undergo separation, or simply we can the separation of the sister chromatids of homologous chromosome. As a result, each cell receives only one gene or what is called an allele. Receives only a single factor or a single allele of a gene for a particular character. So as a result, we see that from meiosis we are receiving 4 cells. The 4 cells are nothing but the gametes, we can say, because meiosis occurs only during the gamete formation. So what is happening during anaphase 2? The separation of what is called the sister chromatids of the homologous chromosomes. As a result, each cell receives only a single allele for a particular character. So we are getting 4 different types of gametes. Each one is having only single allele, either A or small a. So these are things what happen during meiosis. You have to remember these things also. So one, the alleles are present in a particular locus. Then we have the duplication, replication of the chromosomes. And the next one during anaphase 1, the separation of homologous chromosomes. They move to the opposite poles. So we have only the formation of 2 different alleles. The 2 different alleles are separated. Then we have the separation, or what we have the segregation of, or the division of the centromere, leading to the formation of the sister chromatid separation, resulting in the formation of 4 different types of cells, and each cell is having only one gene for a character. So these are all things you have to remember before just considering the behavior of the chromosome. These are all things already we studied. We have to repeat the same one. So now such a diagram is available in the book. Just simply recall that one I represent in a simple way. Normally crossing or occurs, we are getting different things. Here I represent only in a simple way how many cells are produced as a result of what is called meiosis and what is happening, how to recall all these things. Okay, we'll continue further. Scientists established that The number of chromosomes is always constant in cells of a species. Each species has its own chromosome number. Now the highest number of chromosomes is present in one what is called just actually that is gymnosperm, what is called ophioglass. Now the sporophyll is looking like what is called actually the snake's tongue. That's why the name is given as ophioglossum. Ophio, that is snake, glossa, the tongue. That's a sporophyll. That is looking like adder's tongue. That is actually a person. Now it has the highest number of chromosomes of 1262. Then we have Echisitta, that is horsetail, a fern. It contains about what is called 216, the number of chromosomes. Sugarcane, that takes about 80 chromosomes. Rice, we have 24. If you are taking potato, I didn't mention here, it has nearly 48 chromosomes. Then if you are taking maize, it contains about 20 chromosomes. Then we have onion. In the case of onion, we have what is called 16 chromosomes. If you are taking wheat, that contains 14 chromosomes. Okay, so like that we have. That's about the nature. Now the least number of chromosomes is present in Haplopappus glacialis. It is nothing but a slender golden weed having the least number of chromosomes of 4. Okay, even in the case of, for example, 1, Thailcrus. This is a common name. It is belonging to the Astraceae family. Now this one, Haplopappus glacialis, also belongs to the Astraceae family. And this one is called Arabidopsis. Arabidopsis thaliana. It has 10 chromosomes. Now it has been taken for what is called genetical study. The most important plant that has been taken for genetic study belongs to the Cruciferae family. They have the petals in the form of cross. So Arabidopsis thaliana. Even also Human Genome Project, just like that one, the Genome Project has been completed in the case of Thailcrus, Arabidopsis thaliana, 10. Now we have in the case of Haplopappus glacialis, the least number of what is called chromosome, that is a 2 pairs only. It is a slender golden weed. And we have different chromosome number, even in the case of, say an example of golden pea plant, 14. In the case of wheat, also 14. So like that we have. So in the case of cotton, 26, like that we have the different chromosome number. Potato, 48 mentioned. So there are so many numbers, but in the book given only a few. Just I mentioned only a few. You have to go through the table column, what I am going to mention in the video. Okay, chromosome number and difference. Why I am telling that one, the chromosome number is constant for a particular species. You have to remember the highest number of chromosomes in what is called Ophioglossum. That is nothing but a gymnosperm. And also the lowest number of chromosomes, that what we have, Haplopappus glacialis, commonly called as slender golden weed. Okay. While Sutton and Boveri actually made or proposed the chromosomal basis of inheritance, that concept was experimentally proved in 1910 by T.H. Morgan in Drosophila melanogaster. So he was awarded Nobel Prize in 1933, either for physiology or for medicine, for just discoveries concerning the role played by the chromosomes just in heredity. So Nobel Prize winner, he made a number of contributions to genetics. Okay. Now, what are fossil genes? Now we have nearly 98% of our DNA is called junk DNA. So some part of that junk DNA means not usable. They do not have any information for protein synthesis. 98% of human DNA is only junk DNA. We have only 2%. Whereas in the case of Arabidopsis, I mentioned about what is called Thailcrus. It is having nearly 50% of the DNA, what is called specific DNA with the genes, and more than 50% a workable gene. But we have only 2% of the genes only workable. So the junk DNA, the one which normally doesn't have the information. So some part of the junk DNA contains pseudogenes, the false genes. That means these genes were normally working in the past, just like the vestigial organ, now not working. In the case of vestigial organ, they were present efficiently in our ancestors, but now reduced and functionless. The same manner, those genes which are not functioning now, but were working efficiently in the past, they were called as the pseudogenes. So the sequences present in that pseudogene was once working genes. That means we can say the gene is present without function, functionless genes. And they are called pseudogenes, and they contain sequences, and these sequences were working in the past, not now. Now as a result, the pseudogene lost the ability to make proteins. The main function of the gene is its expression to form proteins. That ability is being lost. Simply they are present in the junk DNA, non-use of what is called non-usable DNA. And though present, they are not working, hence called as pseudogenes. Now, but they tell the story of evolution through the fossilized genes. These are also called fossilized genes, you know. The remains of the past. The pseudogenes are present once upon a time, now also present but functionless. They were working, and they normally tell the story of evolution through the fossilized genes. And such genes are called fossil genes, okay, present in junk DNA. That's about a few words about T.H. Morgan and the fossil genes. Now let us have a concept, linkage. What is linkage and how has it happened, etc. when it is possible. Normally, the genes which are the determinants of characters are carried on the chromosome. The genes for different characters may be located on the same chromosome or different chromosomes, what is studied already in the classical genetics. Now, when the genes are present in different chromosomes, they are sorted out independently according to the Mendel's law of independent assortment. So, independent assortment is possible only when the genes are present on different chromosomes. Okay, according to the law of independent assortment of Mendel. But biologists, they came across certain genetic characters did not actually, that is, assort out independently. So, they have found some deviations from what is called the independent assortment of Mendel. You know, Mendel did not know the phenomenon of linkage and crossing over. He was lucky enough because the genes what he had chosen are present in different chromosomes. They assort independently and unite at random, getting 4 different types of gametes without crossing over. But here you see that what they observed, biologists observed, in certain organisms, the genetic characteristics did not assort independently. In other organisms, after Mendel's work, so one such deviation was observed by William Battison and Reginald C. Punnett. Sorry, spelling is there. William Battison and Reginald Punnett observed a deviation from independent assortment of Mendel's law in what we have sweet pea Lathyrus odoratus. They crossed 2 varieties of plants. So it is a dihybrid cross. They have taken 2 pairs of characters are involved. One pair of characters is related to the flower color, another pair of characters is related to the shape of the pollen grain. 2 pairs of characters, so it is called a dihybrid cross as a rule. Now, they took a homozygous strain of purple flower with long pollen grain. So one character, flower color, another character, the shape of the pollen grain. Then another strain of homozygous individual for 2 characters. So round flower with what is called actually, so red flower with a round pollen grain. Red flower with a round pollen grain. So the flower color is red, the shape of the pollen grain is round. They crossed. In F1 generation, all the progenies had what is called purple long character, indicating that the purple flower is dominant over red flower and long pollen grain is dominant over what is called actually the round pollen grain. So we have received the genotype. See that one. This is a heterozygous genotype. We will discuss in later best. We have a separate what is called the topic for linkage. I have given only a simple one, what it's not given in the book. So all the individuals, all the progenies obtained in F1 generation had that is what's called purple flower color with a long pollen grain. But it is a heterozygous individual. So they effected a test cross. What is test cross? You know that one. Crossing of F1 hybrid with the homozygous recessive parent. In monohybrid cross, we are taking only what's called homozygous recessive parent. Here, two characters are involved. We are crossing a parent which is normally homozygous double recessive. Homozygous double recessive. Okay, we have homozygous double recessive. So, an example, this is homozygous double recessive character. The gene homozygous double recessive. And here we have what I mentioned just already. We have that is. This is a heterozygous individual. We'll talk about later with illustration. So all the progenies in the F1 had what is called actually heterozygous purple long. When test cross was effected by crossing this what is called heterozygous F1 progenies with the homozygous double recessive parent. As in the case of test cross, you know that the expected ratio is 1:1:1:1. That's why normally it is used to find out the nature of the genotype, what is studied. So in test cross, the expected ratio is 1:1:1. What we have, the purple long, purple round, then red long, red round. This is the expected ratio. But they didn't get the expected ratio. Here you can see they observed the ratio was 7:1:7. Now these two are the parallel characters. These two are the parallel characters. What is called the purple long, red round, because they were taken as a parallel varieties for cross. Now, the parallel combinations exhibited more number than what we have the recombinant varieties. So, increased number of parallel combinations. So, increased number of parallel combinations occurred in the F2 generation. So, they concluded that the genes for what is called the purple flower and long pollen grain and the genes for red flower round pollen grains were found close together in the same homologous pair of chromosomes. In the same homologous pair of chromosomes, one pair of chromosomes, in the same homologous pair of chromosomes. Okay, that's why they are outnumbered. And what is the reason for that one? As the genes were found close together, the tendency of these genes to stay together without assortment. They remain together or they stay together during the separation of chromosomes. They are not being assorted independently. When the genes are present same chromosomes, when they are not assorted independently during the formation or the separation of chromosomes, that phenomenon is called linkage. So we have increased parallel combinations. It's not a complete linkage. Increased parallel combinations than what we have just the new combinations. So that's why they conclude that actually the genes for purple flower color and long pollen grain and the genes for red flower and long pollen grains were found close together, the same homologous pair of chromosomes. That's why we have increased parallel combinations. This tendency of genes to stay together during the separation of chromosomes is called linkage. I represent only the illustration. But we have a different diagram, different explanation. This is one explanation. We have to go for cis arrangement, trans arrangement, etc. I'll discuss the picture, the stick figures. That's about what we call the linkage. Now we have to do just one more paragraph I want to conclude. What is normally linked genes and unlinked genes? What are linked genes and unlinked genes? The genes located close together on the same chromosomes and inherited together are called linked genes. Hey, here I represented a simple picture. You see the two genes which are lying close together. Okay, the genes located close together on the same chromosome. On the same chromosome and inherited together is called, actually these genes are called linked genes. Now what do you mean by unlinked genes? Suppose you are taking a chromosome. I represented a simple picture having two genes which are placed far apart. Okay, and such genes are not linked. They give a chance of crossing over. So the genes that are sufficiently far apart, you see the two genes are placed far apart. I represented G and G representing the genes. They are sufficiently just placed far apart or located far apart on the same chromosome. They are called unlinked genes. They are also called syntenic genes. They are also called syntenic genes. And the phenomenon, the condition is called synteny. How can you differentiate the linked genes from unlinked genes? So it is to be differentiated by the value, what is called value of recombination frequency. Value of recombination frequency. What is recombination frequency? That is a measure of distance between the two genes. So we have, it may be calculated as centimorgan, percentage recombination frequency. We will do that one with problems after just some time, after finishing. Anyway, so you can measure the distance between the genes by means of what's called recombination frequency, simply called as RF. Or we can say percentage recombination, or we can say centimorgan. This is called mapping of genes on a chromosome. We have separate topic later. Anyway, remember, the one which is to identify whether the genes are actually linked or not linked, you can just use what is called the recombination frequency. Suppose if the recombination frequency value between the two genes is more than 50%, the two genes are unlinked, not linked. If the recombination frequency is less than 50%, then the genes are linked. So more than 50% the genes are called unlinked genes. Less than 50% of recombination frequency, we can say what is called the linked genes. Now if the genes are placed closely, that is very close to each other, they exhibit what is called strong linkage. They cannot be separated by means of crossing over. There is no recombination is possible. So closely linked genes normally show strong linkage, whereas the widely linked genes show weak linkage. So crossing weak linkage means actually the genes are placed farther apart, so crossing over is possible. But here the genes are placed closely, tightly, hence no process of crossing over. That's why we can say strong linkage. And the second one, weak linkage, the genes are placed actually farther apart. They allow the process of crossing over. We have later the problems related to I discuss. So I will draw the diagram for this one. Whether I want to conclude, we have to continue further in the next class. Okay, I want to draw the diagram and show the linkage, the unlinked genes, linked genes. Okay. I described about what we have the linked and unlinked genes, the definitions. Now here I just drawn diagrams to show the arrangement of linked and unlinked genes on chromosomes. The first diagram, I represented two genes, 1 and 2, which are closely located together on the same homologous chromosomes, hence called linked genes. Here I am taking a pair of homologous chromosomes. Gene 1 is located in one member and gene 2 is located in another member. It is also considered as unlinked genes. Then what about here? Syntenic genes. So when the genes are placed far apart on the same chromosome, then they are called unlinked genes or syntenic genes. The condition is called synteny. So here when the genes are placed farther apart, they show what is called, that is, weak linkage. So these are all the widely placed genes, they are placed far apart. Here the linkage is very much limited and exhibit what is called weak linkage. So there is a chance of crossing over possible between the two genes. Now here gene 1 and gene 2, they are placed closely. They exhibited what is called the strong linkage. When the genes are placed close together, they exhibit what is called the strong linkage. So here there is no chance of crossing over between genes 1 and 2. They are possible between gene 1 and 2. So that's about what we have, the linked genes, unlinked genes, unlinked genes. Here this is between, even when the genes are present on two different members of a homologous chromosome, they are also called unlinked genes. But we are normally considering one of the genes on the same chromosome for the phenomenon of linkage. We are leaving this one simply for diagram as given in the book. So genes present actually close together, genes present farther apart, far apart. So they exhibit unlinked conditions and this one exhibit linked conditions. Okay, this one show what is called strong linkage, that one shows what we have weak linkage. We'll discuss in more and more information about these things in the coming classes and the forthcoming classes. Okay, till then, goodbye, thank you.